Assalamualaikum students, I am Nayab Idrees and I am your biology teacher from here on out. We're going to start in the name of Allah, the most merciful and the most compassionate. Today we're going to do the topic transport in mammals. So what is basically transport in mammals? It's the transport system in mammals which transports substances in and out of the cells in a mammal. So first we're going to talk about why do we even need a transport system? As in unicellular organisms are organisms which have only one cell in them. So in unicellular organisms, the demand of transportation can be met by simple diffusion. So there's only one cell, as you can see in the diagram. This is the unicellular organism called amoeba. So in a unicellular organism, there's only one cell, and simple diffusion can be very easy, and it can meet the demand of transportation of substances between it and the environment. If it wants to get substances from the environment, and substances diffuse into it, and if it just has to release the products, the products will just diffuse out of the amoeba and go into the environment. So this is very easy, and diffusion can handle it, and diffusion can meet the simple transportation of a unicellular organism. But when you talk about multicellular organisms which have a lot of cells in them, multicellular organisms cannot use simple diffusion to meet the demand of transportation. Why? Multicellular organisms have a lot of cells, and all the cells can't use only diffusion to transport substances in and out of them, because that'd be very slow and that won't work properly, and it won't be an efficient system of transportation. So hence, multicellular organisms need a proper system of transportation. So as we saw that multicellular organisms have a lot of cells, and diffusion can't meet the demand of diffusion can't meet the demand of transporting substances in and out of cells. So there is a proper system of transportation to transport the substances in and out of cells. Let's go to the next slide here. So the only thing that transports substances in and out of cells is blood. It has a lot of things in it which are transported into the cells and some things out of the cells. So we're going to see the composition and the composition of the blood and detailed composition, and we're going to see what it has in it. So blood is made up of plasma, blood cells, and platelets. These three are the main parts of the blood: plasma, blood cells, and platelets. Now, there are two types of the blood cells called the red blood cells and the white blood cells. Plasma, blood is made up of plasma, blood cells, and platelets, and blood cells have two types called the red blood cells and the white blood cells. Here you can see these are the red blood cells, and these are the white blood cells. You can see these are in red in color and they are purplish in color. Now let's go to the other slide. So we're gonna read about detail of composition of the blood. So first, as we saw, plasma. Plasma is a pale yellowish liquid, pale yellowish mixture. It is a mixture of various soluble proteins such as fibrinogen, and there's some other proteins called albumin and globulin. So there are many proteins in the plasma. There are dissolved mineral salts in the plasma, and there are excretory products which have been taken out from the cells, and there are hormones which are secreted by the glands. Here you can see this is the whole structure, and over here you can see there are plasma molecules, and there are red blood cells and white blood cells, and this is a vessel and the blood is going. You can see this diagram. Now, plasma is a yellowish liquid which is present in the blood, and it maintains the fluidity of blood. That means that if plasma hadn't been over there, the blood wouldn't have been fluid, it would be just solid, blood cells and others. So plasma has to be there, and it also has soluble proteins which help in many things. It has excretory products which have been taken out of the cells, and dissolved mineral salts and hormones. Now, let's talk about platelets. Platelets are small, colorless, disc-shaped cell fragments without a nucleus found in large numbers in blood and help in clotting. So the platelets are not actually cells. They're just like parts of the cells. The cells are fragmented, but they do have a nucleus, but they don't have full cytoplasm, so you can't say that they're cells. So they're found in large numbers in blood, and they also help in clotting. Clotting is a process when the blood flows and the platelets stops the blood from flowing. And you can see these are platelets, these white cell, not cell, but white things you can see associating the red blood cells. Now we're going to talk about the blood cells. The red blood cells, also called erythrocytes, are circular, flattened, and biconcave. Circular means that they're circular in shape. They're like sphere type things. They're circle. And flattened means they're not like inflated. They're just flat. And biconcave means that if we cut them, there's a cross section and it looks like a concave lens. Means that it is bulged in. You can here also see over here in this diagram that the red blood cell has been halved, and you can see that its shape is like this, like it's concave in it's like in the inside. So that is biconcave. It doesn't have any nucleus, even though it's a cell, but it doesn't have any nucleus. That's why it it is worn out and destroyed quickly. Its lifetime is like 3 to 4 months. That's very small. So that's why they don't have nucleus. That's why their lifetime is very small. Red blood cells can squeeze through capillary walls. They don't have a nucleus, so they can squeeze through capillary walls easily. And they also change their shape a little bit when they're squeezing through the capillary walls. Produced by bone marrow, they are produced by bone marrow. The bone marrow, which is the substance which is present in bones, it is produced by that. It has a protein called hemoglobin. The hemoglobin carries out oxygen, and hemoglobin is in red in color, and when it joins with oxygen, it makes oxyhemoglobin. And hemoglobin produce iron and bile pigments when broken down. When hemoglobin is broken down, digested, it produces iron and bile pigments. Bile pigments are the pigments of a specific digesting liquid which is present in the liver called bile. So there are bile pigments and iron and hemoglobin. When worn out, destroyed in spleen or liver. When the red blood cells are worn out, means they're like destroyed and they have to be repaired, but they destroy in the liver or the spleen. The diameter is less than 0.01 millimeter. White blood cells called leukocytes. So they're usually larger than red blood cells and fewer in number because they're larger, so they're fewer in number, and they have irregular shapes containing nucleus. They do have nucleus, but their shapes are irregular, not like circular like red blood cells, or they don't have a specific shape. They can be in any shape. So then white blood cells have two types called the lymphocytes and the phagocytes. The lymphocytes produced by lymph nodes. Lymph nodes are present in the lymphatic system. The lymphocytes are produced by the lymph nodes, and they also produce antibodies, lymphocytes. Lymphocytes show limited movements. They don't move a lot. They're very slow, and they don't have a lot of movements. Phagocytes produced by bone marrow. The phagocytes are also produced by bone marrow, just like the red blood cells. Phagocytes ingest bacterium. They protect the body by ingesting the diseases or bacterium that has entered the blood or the body. Phagocytes have two types called monocytes and polymorphs. So monocytes have bean-shaped nucleus. They have bean-shaped nucleus, and polymorphs have nucleus with many lobes, and it has non-granular cytoplasm. So these are the differences between monocytes and polymorphs, that monocytes have bean-shaped nucleus, while polymorphs have nucleus with many lobes and non-granular cytoplasm. Now we read all the composition of blood. Now we're going to read the functions of blood, like what does blood even do? Acts as a transport medium for oxygen, excretory products, and digested food. Oxygen that is taken out in the lungs, it helps in the transportation of oxygen. And excretory products, which have been taken out of the cells and they have to be supplied to the excretory organs, so it also transports the excretory products. And digested food substances, which are supposed to go to the cells after digestion, so it also transports the digested food substances. It also plays a role in protection how it clots blood when blood is flowing. It also like blood clots itself. How does it do that? So there's an enzyme present in the blood called thrombokinase and thrombin. So the thrombokinase activates the prothrombin into thrombin, and as I told you before, it has a protein called fibrinogen. So the thrombokinase activates the prothrombin to become thrombin, which activates the fibrinogen into fibrin. And when it is soluble and it is activated, fibrin becomes a thread-like structure which entangles the blood corpuscles and clotting occurs. So the fibrin has a thread-like structure which entangles the blood, like they are entangled and they can't move. So the blood cells are entangled and they can't move very rapidly, so the clotting occurs. So this is a protection, because if the clotting wouldn't have occurred, the blood would just not start flowing and we'll lose blood from our body. Blood also produces antibodies and phagocytes, which I told you before that they ingest the bacterium which has entered the body or the blood. Now, organ transplant and tissue rejection. So, organ transplant, when a person's organ is failed or damaged, it is replaced by another healthy organ. So, as in the diagram, you can see that if a person somehow the organ is diseased or damaged, or the failure occurs, so it is replaced by another healthy organ, but there are some conditions for replacing the organ. So as tissue rejection occurs when the organ which is to be transplanted is rejected by the person's immune system, which means that the tissues in the organ do not match with the recipient or the person who's getting the organ, and it is rejected by the immune system. Means that the immune system thinks that it's a bacterium or it's a disease or it's something, a foreign threat or a foreign body, and it produces antibodies to damage it and kill it. So then it won't be any advantage because the immune system would just damage the organ. So this is called tissue rejection, when the immune system treats the organ, the transplanted organ, as a foreign body and produces antibodies to damage it. Now, prevention of tissue rejection. How can we even prevent tissue rejection? How can we prevent tissue rejection by use of drugs which suppress the immune system, and the immune system stops working properly, so the transplanted organ is not rejected. So there are drugs which suppress the immune system, means that the immune system stops working, so it can't produce antibodies and phagocytes or lymphocytes, which when the organ is then not damaged and the organ is not rejected, then because the immune system stops working. But there are some problems with it, that the problems would be that the person won't be immune to minor diseases. If the immune system stops working properly, it can't even defend in minor diseases. So that be a problem. So the person will have to get treated the rest of his life. Second is that the tissue or the organ to be transplanted should be as genetically close as possible, so chances of rejection are low. So when tissue or tissues or organs which are to be transplanted should be genetically close, so tissue rejection is low, because it's like you can take your organ or tissue from your brother, sister, parents, and close relatives, so your genetic information would be as close as possible, which will prevent tissue rejection, as the immune system won't treat the organ or the tissue to be a foreign body because it's genetically close. Weakening the bone marrow, which produces the lymphocytes because they help in making antibodies. So the bone marrow, as I told before, and they produce the phagocytes and lymphocytes, which help in producing antibodies and killing diseases. So when there won't be any lymphocytes or lymphoid tissues which produce lymphocytes, they won't be they won't be helping to make the antibodies, which will slow the process of tissue rejection. So these were some of the points which could prevent tissue rejection. Thank you for coming, guys. I hope you like my lecture, and if you find this video helpful, please subscribe to the channel and like my video. So Allah Hafiz.